Hello everyone. Hello everyone, and welcome to this audio podcast. This audio podcast is part of the conglomerate. This audio podcast is part of a Lobostic Storm. This audio podcast is part of our Storm and Battle Chronicle. This audio podcast is part of the Ztormish series or edition, and this audio podcast is part of Storm Lobostics production. Today, a recap or a look at at uh, sort of a biomarker, an important biomarker, a biomarker to mimic certain disease or condition, certain pathophysiological 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 conditions in around the kidneys. A biomarker referred to as nephrotoxic nephritis or NTN. So a bit of intro. So a research with animal models or through animal investigations have shown the crucial interaction between inflammatory cells and cells intrinsic to the kidney that have been both fundamental and unique to the pathogenesis of glomerulonephritis. Glomerulonephritis being a sort of one of the main sort of pathological conditions where nephrotoxic nephritis have been historically utilized. The terminology glomerulonephritis or inflammatory or inflammation or inflammatory in around the glomeruli encompasses a range of immune-mediated conditions that cause inflammation within the glomerulus and other compartments surrounding renal pathophysiology. The mechanistics of interaction between these cells and the mediators of coordinate of sort of the interlinked coordinated response to inflammation are and have been somewhat elucidated. At the same time, despite the pathophysiological advances, pharmacotherapies, for example, for glomerulonephritis remain non-specific and remain sort of non-specific, and in some cases are sort of hazardous and only partly successful. Therefore, GN, glomerulonephritis, remains a sort of common cause, of CKD or chronic kidney disease globally. In around the mechanisms or mechanistics, evidence of compartment deposition within the glomerulus is a main feature of many forms of GN, and the role of complement is implied by several observations in human disease or human condition in amongst the human mechanistics where the mechanistics have anomalies in around a GN. Indirect evidence obtained from human investigation has been supported or evidenced by findings in animal laboratory investigations. The mechanistics by which macrophages, for example, mediate renal injury remains to be sort of follow up and confirmed, but it is likely or has been likely to include the promotion of inflammation through release of pro-inflammatory cytokines such as tumor necrosis factor or TNF, and recruitment of macrophages and other leukocytes through release of chemotactic molecules. Chemotactic molecules include monocyte chemotactic protein 1 or MCP1, and cell proliferation by a release of growth factors such as macrophage colony stimulating factor or MSCSF. CSF have been established through evidence guidelines and amongst the literature. Cell death at the same time occurs through the release of reactive oxygen species, including nitric oxide, and control of scarring or fibrosis, and repair through release of various enzymes. And as part of the epidemiology, population-based screening investigations have shown that evidence of renal damage by hematuria and low calculated glomerular filtration rate, or a combination of these features, is present in just under around 20% of in amongst adult populations, and similar in amongst different countries or different regions. Accurate classification requires a histological diagnosis. Consequently, the reported prevalence vary according to local indications for renal biopsy. A retrospective analysis of renal biopsies performed between in around the mid 90s, in an Australian population, have found that in around 22% of the population per 100,000 population per year underwent a renal biopsy, yielding an annual incidence of biopsy proven GN or glomerulonephritis of in around 12.3 per 100,000 population. The most commonly diagnosed types of GN in adults amongst adults had been IgA nephropathy, focal and segmental glomerulosclerosis, and vascularitis. Those most commonly diagnosed in children or young people with minimal change disease, focal and segmental glomerulosclerosis, and lupus nephritis, and IgA nephropathy. In around the pathogenesis of GN is complex. Intricacy arises from factors such as autoimmunity, cancer, and structural abnormalities within the renal pathophysiology. There is variability in the susceptibility to GN, which is likely to have a genetic basis, and there are also complex interactions between soluble factors, including for example, including antibodies, complement chemokines, cytokines, and growth factors, and cells where both leukocytes and intrinsic renal cells that mediate glomerular inflammation. The contribution of non-specific factors such as abnormalities in ABP, arterial blood pressure, promote continuing renal scarring or damage despite resolution of an initial insult. The constant feature of GN is the presence of inflammatory leukocytes within the glomerular and the interstitial compartments surrounding renal pathophysiology. Macrophages and T lymphocytes are the dominant dominant cell types present and the intensity of cellular accumulation, particularly within the interstitial, rather than the glomerulus, which has repeatedly been found to correlate with the clinical and pathological severity of disease or condition in animal models, animal investigations, and in human beings. Excuse me. Now looking in around animal models, and in around nephrotoxic nephritis. So animal models are used or utilized during the research and investigation of human diseases or human conditions, and the animals selected will usually meet determined taxonomic equivalency to humans in order to react to disease or its pharmacotherapy or treatment in a way that resembles human pathophysiology. Secondly, research relating to NTN, GN can be traced back to the 1960s and the late 1970s, when observations were reported on the role of complement in NTN produced in laboratory rat investigations by either in rat laboratory investigation, but additionally by the use of either rabbit or duck nephrotoxic sera. Thirdly, in addition, the course of complement reactions in the glomerulus in nephrotoxic nephritis is used as a basis of the course of immunological events in the immunological cascades in glomerulonephritis or GN. Additionally, animal investigations of GN closely replicate features of human GN, have provided insights into the disease, the reproducibility and uniformity of such animal models has also permitted a step-by-step dissection of many of the mechanistics involved. Additionally, again, nephrotoxic nephritis or NTN is produced experimentally by the intravenous injection or infusion of antibody directed against glomerular basement membrane or GBM antigens of recipient species. Most of the accumulated inflammatory cells in many forms of human and experimental GN are macrophages. In amongst animal models, and in specific, particularly in around laboratory mice, BALBC murine mice models have helped clarify the pathogenesis in several renal diseases or renal conditions, and in a retrospective research, 6-week-old female BALBC murine laboratory investigations were induced. Laboratory mice were induced with injections of 50 milligrams or 0.2 mil bacterial lipopolysaccharides or LPS from Salmonella twice a week for 5 weeks by and through the intraperitoneal route. In all mice laboratory investigations, features of polyclonal B cell activation or PBA and GN glomerulonephritis had developed. The animal model presents features that are often encountered in human glomerulonephritis or GN, particularly those associated with bacterial infection as well as autoimmune immune diseases such as systemic lupus erythematosus or SLE. Now looking into into a little bit in around animal models, and specifically in around laboratory rat investigations, or through laboratory rat investigations. So the WKY rat strain is also has also highly is also has is also and has been highly susceptible through experimental investigations of crescentic glomerulonephritis, or CRGN, including through NTN and experimental autoimmune glomerulonephritis, or EAG, or EAG. The laboratory rats or investigations show development of NTN phenotypes, including glomerulosclerosis. More recently, CRGN has been induced on anomally resistant laboratory rat investigations, where the genetic background and in around where the genetic background in laboratory rat investigations are normally resistant, and where investigations also have identified Lewis strain on two genetic loci, CRGN1 and CRGN2, as a new and potentially valuable model or modality for an NTN-GN. The nephrotoxic serum nephritis (NTS) and WKY laboratory rat through WKY laboratory rat investigation have also been utilized in research amongst research, and macrophages and T cell lymphocytes are the dominant cell types present, and the intensity of cellular accumulation, particularly within the interstitium rather than the glomerulus, have been found to correlate with clinical and pathological severity of disease or condition in animal models and in humans. Now, a sort of a look at the in around the role of chemokines. The role of chemokines. So the migration of inflammatory cells into an extravascular site, it requires a series of coordinated signals, including the generation of chemotactic or gradient by the cells of extravascular compartment, and the nature of the stimulus or stimuli, and the subsequent spectrum of chemotactic factors produced determine the specific leukocyte population recruited into the inflammatory site. Members of the chemokine family play a role in the process by stimulating specific subsets of leukocytes. Animals genetically deficient in chemokines are resistant to disease induction, although results have not been uniform or unison, with an unexpected attenuation of experimental antiglomerular basement membrane disease seen in laboratory mice investigations, deficient in CCR1, a receptor for rantes and macrophage inflammatory protein 1, or MCP1. Chemokines are thus identified as important in laboratory mice investigations, and are clearly active in human disease. Additionally, intriguingly, there have been investigations where one team generated CXCR3 deficient laboratory mice investigations to determine the functional role of CXCR3 under pathological conditions, NTN had been induced to analyze the molecular mechanistics of T cell recruitment in glomerulonephritis by CXCR3 and its three ligands, IP10, CXCL10, MIG, CXCL9, and ITAC, CXCL11, and to identify potential new targets for pharmacotherapeutics. The research had identified that chemokine receptor CXCR3 sort of mediates renal T cell recruitment and subsequent tissue injury in NTN. The investigation provided a rationale for considering CXCR3 blockade in T helper 1 mediated renal inflammation as a future pharmacotherapeutic option for recipients with GN and GN subsets. Finally, as part of this audio podcast, a sort of a summary. So, future efforts are required to translate animal investigations in order to achieve a better understanding of disease or conditions in humans. The complexity of GN research, however, makes findings a challenge to replicate consistently. Finally, there must be a sort of continuing evidence base in around the immune and non-immune pharmacotherapeutics in order to provide off bench to bedside practices for those with particular forms of GN, and or particular forms of other sort of challenged healthcare conditions. That draws this audio podcast to a close. This audio podcast is part of the conglomerate. This audio podcast is part of a Labosnik's Shystorm. This audio podcast is part of a Storm Battle and Chronicle. This audio podcast is part of the Ztormish edition or series, and this audio podcast is part of a Storm Labosnik's production.